Assalamu alaikum, ladies and gentlemen. Good afternoon. I am Saddam Hussain, PhD scholar from Government College University, Faisalabad. It's an honor for me to be the moderator for fourth session of Third International Conference on Applied Zoology 2020 on the behalf of organizing committee of Third International Conference on Applied Zoology 2020. This session comprises the fields of natural products, cancer biology, epidemiology, and recombinant DNA technology. Let's start the session with presentation of Honorable Dr. Muhammad Tayyib from University of Veterinary and Animal Sciences, Lahore, entitled Recombinant DNA Technology for the Production of Industrially Important Enzymes. Please, Dr. Muhammad Tayyib.
Bismillahirrahmanirrahim. First of all, I say welcome to all of you in this online conference. And I am Dr. Muhammad Tayyib, serving as Associate Professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. And the topic for today's talk is recombinant DNA technology for the production of industrially important enzymes. So here you can see actually the view of University of Veterinary and Animal Sciences, Lahore. So let's start with the issue. Actually, we are facing some problems in the poultry sector. And poultry sector is the fast growing industry of Pakistan that is committed to provide high value proteins to the nation. But this industry is facing actually the problem, and that is actually feed, the cost of feed. Feed is quite expensive, and due to this expensiveness of the feed, the rate of poultry is getting high. So there are two options. Number one, and we can go to animal-based feed, and number two, we can go to plant-based feed. In case of animal-based feed, that is quite expensive. Whereas in case of plant-based feed, there are certain components of feed which are not being hydrolyzed by the poultry due to the deficiency of enzymes. So these components might include cellulose, xylon, proteins, phytate. We need certain enzymes actually. The supplementation of feed with these enzymes that is actually results in order to increase the growth rate of the poultry bird, and enzymes are actually environment friendly. So simply the solution is to produce the recombinant enzymes at a domestic level, and this production of recombinant enzymes for the fulfillment of industrial requirement at local level, that will actually cause to save the huge foreign exchange. So what is actually this technology? This technology, which is called recombinant DNA technology, that has actually a few steps. Those are in front of you. You can see we need genomic DNA, and the genomic DNA will act as a source for providing your required gene of interest, right? And then this DNA can be utilized for its amplification to increase the copy numbers via the PCR, and the amplified PCR product will be utilized for its cloning in the cloning vector. The purpose of cloning in the cloning vector is to increase the copy number. Then this gene from cloning vector can be shifted towards the expression vector. Once the gene is in the expression vector, then the process of as a result of process of transformation, we can have the transformants. Now these positive transformants can be utilized in order to confirm the availability of gene in the vector. And secondly, after the process of expression, now we have the protein, recombinant protein or recombinant enzymes. These enzymes, after the process of expression, these will be purified and will be utilized for the characterization. And once the characterization process has been completed, then the enzymes can be utilized for the supplementation of poultry feed, and when that feed which is being supplemented with the enzymes, recombinant enzymes, that is being utilized in order to feed the poultry bird, then ultimately you can find a positive impact of available enzymes on the weight gain in the birds. So we have characterized actually a number of different enzymes which are important for the supplementation of poultry feed. So briefly and very quickly, I want to share with you actually the results for a recombinant phytase, which was actually from Thermotoga naphthophila, a thermophile. And you can see the purified protein, that is phytase. Its optimal temperature 80 degrees Celsius, then thermostability studies, optimal pH, it was between 6 to 7, and then effect of metallines, kinetic studies. Similarly, the characterization of recombinant cellulase, its purified protein, its characterization studies for temperature, thermostability, pH, metallines, detergents, and kinetic behavior of this. In the same way, a recombinant xylanase that was actually characterized, you can see its purification, effect of temperature, pH, metallines, detergents, and its kinetic studies. Then protease, this protease was actually from Geobacillus SPS4S. You can see its purified form, and then temperature, pH, metalline requirement, detergents, and kinetic studies. So we have actually completed the characterization of these enzymes which are being utilized for the supplementation of poultry feed. Now, the feed was prepared that was supplemented with all of these enzymes separately in order to find the impact on the growth. Now, in future, we have a plan to make a recombinant enzymatic mixture, and inshallah, after the preparation of that mixture, we will utilize that mixture for the supplementation of poultry feed. And finally, the results will be examined in terms of growth of poultry bird. So simply the outcomes of this study, that is actually the supplementation of poultry feed with the locally produced enzymes that showed a significant impact on weight gain of poultry birds. And this locally produced enzymatic enzymes in future will be utilized for in order to find its impact on the growth of poultry birds. Actually, we have examined the impact of these enzymes separately on the weight gain, but our goal is, our objective is to prepare an enzymatic mixture and to utilize that enzymatic mixture for our local industry. Thank you.
Thank you, Dr. Muhammad Tayyib, for your wonderful speech. Now I would like to invite our second speaker, Dr. Shugufta Naz. Dr. Shugufta Naz is from Lahore College for Women University, Lahore, and she is going to present with title Prevalence of Different Types of Endothelial Corneal Dystrophies by Age and Gender and Visual Acuity in Punjab, Pakistan.
Assalamu alaikum. I want to say many thanks for giving me opportunity to present my data in third International Conference on Applied Zoology 2020. My name is Dr. Shugufta Naz. I am working as Associate Professor in Department of Zoology, Lahore College for Women University, Jail Road, Lahore, Pakistan. I did PhD on the molecular characterization of retinal dystrophies from Center of Excellence in Molecular Biology, University of the Punjab, Lahore. Today's the topic of my presentation is the prevalence of the different types of the endothelial corneal dystrophies by age, gender, visual acuity in Punjab, Pakistan. The visual system constructs a mental representation of the world around us, and the cornea is dome-shaped front portion of the eye where light is focused. In ophthalmology, the corneal dystrophies have typically referred to a group of inherited disorders that are usually bilateral, symmetric, slowly progressive, and not related to environmental or the systematic factors. And on the basis of the histological, anatomical histological classification, corneal dystrophies can be classified on the basis of three layers of cornea: epithelial corneal dystrophies, stromal corneal dystrophies, endothelial corneal dystrophies. And the current study is on the prevalence of the endothelial corneal dystrophies. Endothelial corneal dystrophies characterized by a slowly progressive endogenous degeneration of the corneal endothelium, and are at least in a part of it due to the genetic predisposition. And the genetic predisposition is actually the development of the phenotypic development of the individuals of the same species or the population due to certain environmental conditions. The parent study was conducted to find the occurrence of different types of endothelial corneal dystrophy, which include Fuchs endothelial corneal dystrophy, posterior polymorphism corneal dystrophy, congenital hereditary endothelial dystrophy, X-linked corneal endothelial dystrophy with respect to age, gender, in Punjab, Pakistan. The patients suffering from different types of endothelial corneal dystrophies, these were identified with the help of the ophthalmologist, and the performers were given to these ophthalmologists. So the different variables, the data that was collected with the help about these patients of different endothelial corneal dystrophies, this was collected from these with the help of these ophthalmologists. These were analyzed, different variables were analyzed, and after analyzing these variables, these results were analyzed by chi-square application. And among the results, out of 198 cases of the endothelial corneal dystrophies, 38.3% were of the Fuchs endothelial corneal dystrophy. And among this prevalence, 55.26% were males and 44.74% were females, as this can be seen from this graphical representation. However, in the case of the congenital hereditary endothelial dystrophy prevalence, it was 25.25%, and among this prevalence, 64% were males and 36% were females. However, for the posterior polymorphic corneal dystrophy, prevalence was 29.79%, and among this prevalence, 42.37% were of males and 57% were of females. And lastly, the fourth type of the endothelial corneal dystrophy, prevalence of X-linked endothelial corneal dystrophy, that was 6.57%, and among this prevalence, 61.5% were of males and 38.5% were of females. As the basic aim of this research was to determine the prevalence of the different types of the endothelial corneal dystrophy in the population of the Punjab. And among 3,000 patients, 6.6% cases of the endothelial corneal dystrophy were recognized, which includes more males, 52.52%, as compared to the females that were 47.48%. Maximum cases of the endothelial corneal dystrophy, these were 31.82%, were observed in the age group of 41 to 50. In all types of endothelial corneal dystrophy, the Fuchs endothelial corneal dystrophy was observed mostly in the 38.38%. And least cases were of the X-linked endothelial corneal dystrophy that were 6.67%. The prevalence of the congenital hereditary endothelial dystrophy and the posterior polymorphism corneal dystrophy was 25.25% and that of the posterior polymorphism chondrodystrophy, that was the 29.79% respectively. And so from this research, from the results of this research, we can finalize that posterior chondrodystrophy is a very rare disease prevailing in Pakistan with a proportion of only 6.6%. And as its prevalence is very low, and we have visited when we visited these hospitals with the help of different ophthalmologists, we have collected the data of 3,000 patients, and it was proved that it is very rare. But being a geneticist, I can say that they didn't have the idea about the cornea transplantation. That when we visited them and we did the counseling of these patients, and we gave them awareness about the this corneal disease, and so now they have the idea after the cornea transplantation, their vision can be restored. Thank you.
Thank you, Dr. Shagufta Naz, for your informative talk. Now I would like to invite Dr. Muhammad Asif Zahoor from Government College University, Faisalabad to present his presentation with title Occurrence of Multi-Drug Resistance Mycobacterium Tuberculosis from Patients of Pulmonary Tuberculosis in District Khanewal. Please, Dr. Muhammad Asif Zahoor.
My name is Dr. Muhammad Asif Zahoor. I'm working as Associate Professor, Department of Microbiology, Government College University, Faisalabad. And I'm presenting occurrence of multi-drug resistant Mycobacterium tuberculosis from patient of pulmonary tuberculosis in district Khan. According to the WHO, tuberculosis is one of the deadliest infectious disease which is prevalent across the world with high morbidity and mortality, and among this, pulmonary tuberculosis is most prevalent with the highest transmission rate. According to the WHO, 10.4 million people were infected with tuberculosis, and tuberculosis HIV co-infection were 1.8 million. Tuberculosis is the disease which is amongst top 10 causes of the deaths worldwide. Among highest burden countries, Pakistan rank 5th globally. Drug resistance is also a serious issue in the treatment and control of tuberculosis. Unfortunately, Pakistan stands at 6th high burden country in this type of tuberculosis. Annually, almost 0.5 million people, including 15,000 children, were infected in Pakistan. That leads to almost 70,000 deaths. WHO Surveillance Report 2017 indicated that around 0.6 million people have rifampicin resistant tuberculosis, along with 0.5 million patients who developed MDR tuberculosis. And unfortunately, out of these cases, 47% of the patients belonged to India, China, Russia, and Pakistan. The current study was designed to detect the prevalence of pulmonary tuberculosis among the patients attending district headquarters hospital Khanewal, and to determine the relationship of risk factors associated with the tuberculosis, and to determine the frequency of rifampicin resistance among Mycobacterium tuberculosis. For this purpose, a cross-sectional descriptive study was conducted in collaboration of the district headquarters hospital and Department of Microbiology, Government College University, Faisalabad. For this purpose, 200 sputum samples were collected from the patients who attend TB.clinic DHQ Khanewal. The samples were collected according to the guidelines of the WHO. The samples were collected in sterile containers, and the samples were shifted to the Gene Expert laboratory in DHQ hospital. The collected samples were treated with lysing buffer followed by shaking and vortexing, and the samples were allowed to liquefy for 10 to 20 minutes. So here is the processing of the samples. The samples were mixed with the lysing buffer and vortex and immediately shifted to the cartridge, and the cartridge was shifted to the gene expert that worked on the principle of the real-time PCR. And here is the sample report. So first red line indicate the presence of the medium level of Mycobacterium tuberculosis, and the second line is rifampicin resistance among the Mycobacterium tuberculosis is not detected. Here is the one thing is good, that the Mycobacterium tuberculosis level is very low, but unfortunately that low level is rifampicin resistant. And here is the result, that gene expert indicated 80 samples were found positive, and here distribution is high among the males and high among 26 to 50 years age group and high among the rural areas, and it's high in previously treated patients, and it's high rifampicin resistant cases. So in conclusion, the data showed a total of 80 samples which are positive, and this is the epidemiological investigation, and this is increased rifampicin resistance among positive cases. And finally, it is recommended to diagnose the suspected sputum samples using gene expert to ensure accuracy of the treatment. Thank you very much.
Thank you, Dr. Muhammad Asif Zahoor, for your amazing presentation. After such amazing presentation, I would like to invite another young researcher from Government College University, Faisalabad, Amara Riaz, to present his research work with title Screening of Natural Products Against Human Triple Negative Breast Cancer Cells. Miss Amara Riaz.
Assalamualaikum, respected participants and my dear fellows. I would like to start my talk by thanking AZSP for arranging ICOS 2020 virtual conference. My name is Amara Riaz and I am going to talk on the topic screening of natural products against human triple negative breast cancer cells. Cancer is a complicated disease marked by the deregulated epigenetic, metabolic, and molecular signaling pathways. This is one of the most fatal diseases in the world, affecting about 14.1 million people annually. The most dangerous types of cancer include lung cancer, liver cancer, colon cancer, and breast cancer. The topic of my research mainly focuses on breast cancer. This is because Pakistan has one of the highest incidence rates of breast cancer in Asia. This disease kills 40,000 Pakistani women annually. We can see that 1 out of every 8 women suffers the risk of developing breast cancer throughout her life. There are 4 other subtypes of breast cancer, but I have specifically focused on triple negative breast cancer. Now you people may ask that what is triple negative breast cancer? Actually, there are 3 important receptors in breast cancer cells, namely estrogen receptor, progesterone receptor, and human epidermal growth factor 2. In case of TNBC, all of these characteristic receptors are absent, making the situation challenging for the researchers as well as clinical practitioners. So the next question is why we are focusing on triple negative breast cancer. This is because it has high mortality rate, poor prognosis, and there are no available drugs for treatment, as well as it is resistant to currently available chemotherapy. Shortly, we can say that TNBC is a highly aggressive subtype of breast cancer with no currently available chemotherapeutic biomarkers. Review of literature has enabled us to formulate the hypothesis that natural products may have the potential to cure human triple-negative breast cancer cells. Keeping in view this hypothesis, we have formulated the objective of our research. to screen the plant extracts and natural compounds to identify TNBC growth inhibitors using TNBC, that is MDA-MB231, and non-TNBC, that is MCF7 cell line models. To fulfill the objective of our research, we have formulated a screening strategy to determine the anti-cancer potential of natural products against TNBC. For this purpose, we have screened two types of natural product libraries, that is plant extract library and natural compound library via MTT-assay. Plant extract library was established by collecting different type of plants, drying them under shade, and making their extracts using soft slate apparatus and rotary apparatus. Natural compound library was established by collecting different type of natural compounds from customized natural product libraries. The protocol of MTT-assay has also been shown on the right side of this slide. This slide shows the screening results of different plant extracts against human TNBC cells. The graph on the left hand side shows that Mangifera indica seed extract is most potent against human TNBC cells, followed by Mangifera indica bark extract, eucalyptus extract, aloe vera extract, and Calotropis procera leaf extract. Here it is to be mentioned that the plant extract, which inhibited the cell viability by more than 40% at a concentration of 100 microgram per ml, were considered as positive hits. The graph on the right hand side of the slide shows the comparative cytotoxic behavior of these plant extracts. IC50 values of these plant extract has also been shown in this slide. As we have seen that plant extract library, from the screening of plant extract library, we have obtained 5 potent plant hits against human TNBC cells. The next step is the screening of natural compounds against human triple negative breast cancer cells. From our research, we have identified 4 potent plant hits which were found effective against TNBC cells. When these compounds were further screened, we found that 2 natural compounds were selectively effective against human TNBC cells, while the other 2 plants were not selective against human TNBC cells. This slide shows that out of 100 natural compounds screened, only 2 compounds, arioflexin B and resistomycin, the chemical structures of which have been also shown here, were effective in controlling TNBC cells. IC50 values of these 2 compounds have also been shown here. These 2 compounds have been recently reported by me in my research papers. There is also a graphical representation of anti-TNBC activities of ADO-B and resistomycin. Now let me tell you about the novelty or significance of my study. In this study, 51 plant extracts were screened, out of which 5 plant extracts, that is Mangifera indica seed and bark extract, eucalyptus extract, aloe vera extract, and Calotropis procera were found effective against cancer cells. This study is one to report 2 novel compounds, that is arioflexin B and resistomycin, which exhibited anti-TNBC activity. In vivo studies and toxicological profiling are required for further investigations. These are the references I have used for making this PPT. Thank you so much for your attention. Allah Hafiz and stay blessed.
Thank you, Amara Riaz, for such an excellent talk. On our next turn, we have Dr. Sadia Nawaz from University of Veterinary and Animal Sciences, Lahore, with title WNT 10A mutation associated with a complete odonto-onychodermal dysplasia syndrome. Dr. Sadia Nawaz.
Greetings from University of Veterinary and Animal Sciences. I am Dr. Sadia Nawaz, working as Assistant Professor in Institute of Biochemistry and Biotechnology. Today I am going to present a part of my PhD work. Title of my presentation is Wnt10A mutation associated with a complete odonto-onychodermal dysplasia syndrome. Odonto-onychodermal dysplasia is a skin disorder and it comprises a heterogeneous group of developmental abnormalities which constitute about 200 clinical entities. It's a rare syndrome and it's characterized by severe hypodontia, nail dystrophy, smooth tongue, dry skin, keratoderma, and hypohidrosis of palms and soles. The family that we had a surgeon from a Punjab region. We had spent 5 generations in this family, and the clinical presentation of this family showed that the affected members had one of the affected member had sole of right foot in individual 7, and at the age of 37, which was indicating, which was presented with the palmoplantar, with the plantar keratoderma. There were dystrophic toenails and smooth tongue with reduced papilla. It was there, and the panoramic radiograph of the affected nail member at the age of 39 showed that the most of the permanent teeth had third molar excluded, and the upper jaw there are 2 permanent central incisors and 2 permanent premolar with conical crown shape. In the lower jaw, there were 4 permanent premolars with reduced crown form, or the bottom arrows. And the scheme of studies, when we have to study the the inherited disorders, we go for the linkage analysis, which includes, first of all, pedigree analysis, where we can see what is the mode of inheritance or the pattern of inheritance in this family. Then we look for the candidate genes which are linked with that region, and we do the exclusion mapping. And if we don't see any linkage, then we go for the genome-wide screening, and the statistical linkage analysis later again turn to the novel mutations, novel loci, and at times novel genes. Nowadays, NGS has made this much easier than going through all these steps. The family that we had is certain. We spanned 5 generations in this family, and there were 6 individuals which were affected by onychodermal dysplasia, and the inheritance pattern was autosomal recessive. The degree of consanguinity was very high in this family. When we designed primers for the candidate genes, which were tightly linked with the skin disorder, we had found that our gene, our region that was linked, that was associated, tightly linked with this disease was gene Wnt10A. Sequencing of the Wnt10A gene showed the position that was changed, or the transition that we had observed in the chromatogram, that was in the nucleotide replacement C to T, which had ended up in A to V change or replacement. This homozygous Wnt10 emissions mutation that we had seen in all 4 affected cases of the OODD syndrome. The first report of missense mutation, it is that is showing a mutation in the Wnt10A gene, and it has resulted into alanine to valine substitution. Earlier, a Wnt10A nonsense mutation at position 697 G2T was reported in 6 distantly related OODD patients. When we looked at the phylogenetic analysis, the situation was highly conserved across species. To summarize our results, we had observed this with Win10A and W10A mutations in OODD. The homozygous missense mutation is associated with a complete OODD phenotype, contributing to the classification of ectodermal dysplasia. And the findings emphasize the importance of Win10A in the formation of ectodermal appendages such as hair, teeth, skin, and nails in the humans. And the identification of human phenotypes with the other Win10A mutation or possibly mutation in the downstream members of this signaling pathway will be important to further understand the developmental process of the ectoderm. These studies will be improving our current knowledge about Win10A in ectoderm development for both diagnostic and biological importance. The work may prove helpful to study gene function and learning more about the pathophysiology of OOD disease. And in long term, this can lead to a better understanding of the genetics behind ectodermal dysplasia that will contribute to improved diagnostics, risk assessment, therapeutic strategies, and to develop improved treatment models for these disorders. The results of this study will also help to provide genetic counseling to the carrier families for precautions of affected birth, and the study may lead to a marked reduction in the number of severely affected children born with inherited disorders of skin in the Pakistan population. This is the map of consanguinity, and thank you for listening.
Thank you, Dr. Sadia Nawaz, for sharing such information from your research work. Our sixth presentation for this session is by Mudassir Hassan from Government College University, Faisalabad, with title Macrolipiota procera, dietary mushroom as a potent therapeutic agent. Mudassir Hassan.
Thank you very much to the Applied Zoological Society of Pakistan for providing a great platform to present our work in 3rd International Conference on Applied Zoology 2020. I am Mudassir Hassan, MPhil Scholar, Department of Zoology, Government College University, Faisalabad, and today the topic of my talk is Macrolipiota procera, dietary mushroom as a potent therapeutic agent. First of all, introduction to mushroom. Mushroom are highly nutritious and medicinal macrofungi. They are worldwide in distribution. Main component of export of mushroom producing countries like Turkey, Poland, China. Every year these countries produce million tons of mushrooms and export it to all over the world. There are 50,000 species of mushroom, in which hardly 1 to 2% species are poisonous, but all other used for their nutritious and medicinal properties. Nutritional profile of mushroom is very interesting. Include 26 to 82% of carbohydrate, very little amount of fat, 12 to 35% protein. They have a lot of essential amino acids, especially tyrosine, valine, glutamic acid, isoleucine, and also have vitamins, especially vitamin C and vitamin D. It also have micronutrients, which are very important for our proper functioning of metabolism and cellular growth, like selenium, choline. So these mushrooms were collected from Turkey under HEC Park Turk Research Mobility Grant Program, which was titled Targeting the Energy Metabolism with Dietary Nutrient for Cancer Treatment. We collected 50 mushrooms from different parts of Turkey and then make their extract by using green method technology. Why green method technology? The answer of this question lies behind the principle of green method technology. It provides renewable resources, its waste are degradable, it reduces the unit operation It also provides byproducts which are very beneficial. In this method, we also can use alternative solvents rather than CCl4, methanol, and all other toxic solvents. It also reduces our energy consumption and time, which is need of our scientific work. In green method technology, first of all, we wash our mushroom sample, then dry it at 25 degrees Celsius for 3 days. Then we grind our all mushroom samples. Then we did microwave assisted extraction. In microwave assisted extraction, we took 50 gram of sample in 500 ml absolute ethanol. At 400 watt, we repeat 3 cycles. Each cycle was of 6 minutes. Then we get filtrate. This filtrate we then evaporated through rotary apparatus and liofilize to remove water content, and then we get crude extract. We use this crude extract in screening process for all these mushrooms. We did phytochemical analysis, antimicrobial assays, cytotoxic assays, and antioxidant assay. In phytochemical analysis, we check tannins, flavonoid, phenolics, alkaloid, and saponins. In antimicrobial assay, we did antibacterial and antifungal activities. In cytotoxic assay, we check cell viability through MTT assay. And in antioxidant activity, we did DPPH assay to check their radical scavenging activity. By doing all these assay and preliminary screening of these mushrooms, we found Macropiota persera highly potent for all these activities. Here are the phytochemical analysis results of our mushroom Macropiota persera. We found flavonoids, steroids, saponin, alkaloid, and tannins in our mushroom. Antibacterial activity of Macropiota persera was measured against 7 bacterial strains in which 2 are gram positive and 5 are gram negative. The gram positive were Staphylococcus aureus and Pastorella multicida, and gram negative were Pseudomonas aeruginosa, Bacillus cereus, Escherichia coli, Aromonas hydrophylla, Salmonella enterica. The graph shows the zone of inhibition with the concentration in milligram per ml. On the x-axis, here are the concentration, and on the y-axis, zone of inhibition in millimeter. So this shows the maximum activity against Pseudomonas aeruginosa for Macropiota persera. The minimum inhibitory concentration was calculated 187 to 50 microgram per ml, which shows high antibacterial activity of this mushroom against these bacterial strains. Here a picture also shows the activity and zone of inhibition. You can see here clearly. Antifungal activity of Macrolepta persera was also very high. It shows maximum potency against Fusarium evanescens. The other fungus strain, Aspergillus niger and Fusarium brachygibosum, also show almost equal amount of antifungal activity. The minimum inhibitory concentration was in range of 125 to 187 microgram per ml. Here are also zone of inhibition are clearly shown in these plates at the minimum inhibitory concentration given below. The antioxidant activity of Macrolepta persera was very high. It showed the IC50 to 14 microgram per ml with compared to ascorbic acid which chosen as a standard. With the regular increasing concentration of Macrolepta persera, the percentage antioxidant potential also increasing very smoothly. It shows a direct proportion with the concentration. Here are the picture which showing the purple color changes toward the yellow color, which shows the antioxidant potential of Macrolepta persera, and this one is the antioxidant potential of ascorbic acid which chosen as a standard. Anti-cancer activity of Macrolepta persera was also calculated against breast cancer cell line MCF7 cells, and it found to be very potent against this cell line. The x-axis shows the different concentration from 0.15 microgram to 100 microgram, and y-axis shows the cellular viability. By increasing the concentration of Macrolepta persera, the cell viability decreasing. Its direct connection is showing in this graph. And IC50 was calculated 6.18 microgram per ml, which show highly potent against MCF7 cells. Novelty and significance of our result is 50 mushrooms have been collected and extract were prepared through green method technology. All samples were screened different biological activities in which Macrolepta persera was found highly potent for these activities as shown above. It shows MIC less than 125 to 187 microgram per ml against microbial strain, and IC50 value of Macrolepta persera as antioxidant was 214 microgram per ml, which show highly antioxidant properties. Anti-breast cancer activity of Macrolepta persera was also very significant with IC50 6.18 microgram per ml. For future perspective, from above screening data of M. persera could be a novel source of drug formulation against human pathogenic diseases and cancer treatment. It could be a safer drug due to its dietary nature. Thank you for your attention. If you have any query, you can ask in comment box. Thank you very much.
Thank you, young man, for your research presentation. Now I would like to invite Asya Parveen from Bahauddin Zikriya University, Multan, to present with title Molecular Epidemiology of Theileria annulata infection of cattle in Lia district, Pakistan. Asya Parveen.
Third International Conference on Applied Zoology 2020, organized by the Applied Zoological Society of Pakistan and Kaizen University, Islamabad, Pakistan. My name is Asya Parveen. I am PhD scholar from Institute of Pure and Applied Zoology Division, Bahauddin Zikriya University, Multan, Pakistan. My topic is Molecular Epidemiology of Theileria annulata infection of cattle in Lia district, Pakistan. Reason of low productivity of farm animals in Pakistan: non-discrete breed, poor management, lack of nutrition and of resources, inadequate artificial insemination services, diseases and parasitism are the main reason for the low productivity. Tick and tick-borne diseases in cattle affect almost 80% of world cattle population. Ticks are bloodsuckers that not only damage skin, hides and predispose cattle to dermatophillosis and meiosis, induce paralysis, photoxicosis and cause physical damage to livestock. Bovine filariasis or tropical filariasis is caused by protozoan parasite of Theileria species. These are round, wide, rod-like, found in lymphocytes, histiocytes and erythrocytes. Causative agent: Theileria annulata is the main cause of tropical filariasis. Theileria parva cause is fever. Theileria mutant, Theileria orientalis, Theileria mellifera cause milder or non-pathogenic filariasis. Tick vector of bovine filariasis: Exotic tick of genre Rhifis falis, Hyloma, Ableoma, Hemifacialis are the main tick vectors. Hyloma and Itolicum is the main tick vector of tropical filariasis in Pakistan. Life cycle of annulata: Annulata completes its life cycle in two post. Kynete and zygote formation take place in tick, while sporogony and murogony take place in bovine host. Sign and symptoms: temperature may rises up to 40 to 41.5 centigrade. Depression, lacrimation, nasal discharge, swelling of lymph nodes and anemia are the major signs and symptoms of filariasis. Risk factors associated with prevalence of filariasis: climatic condition, tick density, age of cattle, gender, immunity or innate susceptibility, and management practices are the main risk factors. Aims of current project: to report the molecular epidemiology of Theileria annulata in blood samples of cattle. Objective of current project: to study the epidemiological factors associated with the prevalence of Theileria annulata, to report the effect of Theileria annulata infection on cattle, blood profile of enrolled cattle. to report the prevalence of tick infestation in enrolled cattle and their taxonomic identification and detection of parasite in tick to confirm their role as a vector, to sequence the amplified PCR product of Theileria annulata for parasite confirmation, and to conduct the phylogenetic analysis for Theileria annulata by using cytochrome B gene. Material and method. Following questionnaire was filled for determination of risk factor before taking blood sample. Sample number, breed, sex, age, location, management, number of animals in herd, body condition, and history of the trial were considered. Study area and design. 900 samples from 3 different breeds were collected during 4 different seasons. Ticks were also collected and brought to the lab for morphotaxonomic identification. Blood samples were collected and brought to the lab for further analysis. Microscopy and blood smear screening was done to confirm the parasite presence. DNA extraction from the blood was taken by means of inorganic method, while DNA extraction from the tick was taken by means of ammonium hydroxide method. DNA extracted was subjected to further polymerase chain reaction. Component of reaction mixture of PCR, tempered DNA, primer, DNTP, divalent cation, tag DNA polymerase, buffer solution, and CT deionized water was used in reaction mixture. Thermocycler profile of PCR. Initial denaturation 94 centigrade for 5 minutes, annealing 56 centigrade for 50 seconds, final extension 65 centigrade for 5 minutes. PCR products were further subjected to gel electrophoresis to confirm the band size for the Theileria annulata confirmation. Sequence and phylogenetic analysis. Amplified PCR products were sequenced and submitted to NCBI gene bank. Phylogenetic analysis of the amplified PCR products were carried out with the help of our collaborator, Professor Dr. Munir Akhtar, at Prague University, Turkey. Statistical analysis. Data was expressed as mean values and standard error mean. Feature desk tests were conducted to correlate the parasite prevalence with studied epidemiological factors, and 2 sample t-tests were conducted to compare the hematobiochemical parameters. Results. Hyloma species pallis and hemiphysalis are the main ticks identified by different teams during study. PCR amplification represents the C12 base pair for the confirmation of Theileria annulata. Overall seasonal prevalence of Theileria annulata among crossbreed, Friesian breed, and Saigold breed. Autumn was found, the highest prevalence was observed during autumn, followed by winter, summer, and spring season. Overall breed-wise prevalence of Theileria annulata among crossbreed, Friesian breed, and Saigold breed. Crossbreed was found to be more susceptible for theileriosis, followed by Saigold and Friesian breed. Comparison of complete blood count parameters of blood samples of crossbreed cattle. Monocytes, mean cell volume, mean cell hemoglobin, mean corpuscle hemoglobin concentration, and platelets are significantly affected blood parameters. Association of studied epidemiological factors of crossbreed cattle. Sex, water sources, presence of dog at the farms, and tick load on the dogs are the main epidemiological factors associated with the theileriosis. Cytochrome B sequences from Theileria annulata isolate submitted to gene bank under the section number 44, 45, and 46, and the comparison represented the same sequences and 99% similarity of sequences with Iran, China, and Turkey. Conclusion. We recommended this PCR protocol should be used for the detection of Theileria annulata in livestock for their proper diagnosis and treatment. Phylogenetic diversity would be helpful for understanding the distribution and epidemiology of Theileria annulata and for designing the effective strategies to control the disease.
Thank you, Asya Parveen. I hope you viewers are getting a lot from this session. So I would like to invite another researcher from University of Faisalabad, Amar Palwasha, to present her research work with title Evaluation of Rice Husk as a Substrate for the Growth of Lactobacillus Species. Amar Palvasha.
Assalamu alaikum. My name is Amal Palvasha and I'm a student of Amphib Microbiology. And today's my topic of presentation is evaluation of rice husk as a substrate for the growth of Lactobacillus species. What are the probiotics? Probiotics are the live microorganisms which provide the health benefits on consumption and generally by improving and restoring the GI tract. And what are the prebiotics? Prebiotics are the fermented ingredient in the gastrointestinal tract which is not digested by the human intestine, but these are consumed and by the microflora. And the microflora are the probiotic organisms. There are different types of prebiotics like non-starch, starch, polysaccharides, oligosaccharides, amino acids, proteins, and dietary fibers. And these components cannot be digested by human small intestine, but these are fermented by the colonic microflora and they provide the health beneficials to the GI tract by providing the nourishment to the probiotics. And one of the most promising prebiotic is xyloligosaccharide, which is obtained from the plants. These are the main component of the lignocellulosic biomass. FOS and the inulin, these were the best and the promising prebiotics before the XOS, but the XOS shows the temperature and the acid stability rather than the FOS and XOS and inulin, so it is considered as the best prebiotic. And in my research, I used these material method and I procured the rice husk, which is used as a renewable resources to obtain the prebiotic XOS, which is xylo-oligosaccharide. It is an oligomeric sugar. And after the procurement of the rice husk, these were undergone mechanical treatment like sieving, milling, etc. And the water extraction was done in the Stocksley apparatus. Ethanol extraction was done in Stocksley apparatus, and then the acid extraction. After the acid extraction, neutralization was occurred, and after the neutralization, the component and the extract which was obtained from the rice husk was quantified using the HPLC and the HPLC column. And the determination of the rice husk extract was done by procurement of the lactic acid bacteria. I used different kind of species like Lactobacillus rhamnosus etc. and different other species of lactic acid bacteria like bifidobacteria, and these culture were obtained from the culture bank. And these culture were then fermented with the XOS. And after this, the evaluation of XOS was determined. How this process was done, and the XOS was obtained by different processes like chemical, enzymatic, and hydrolytic. Rice husks were the cheapest source to obtain the XOS for the production of the XOS, which is a prebiotic. And so this is how the rice husk, which is used as a renewable resources, these were air dried, milled, sieved, placed in the screw cap bottle in the water bath. Extractive free rice husks were obtained by using the Stocksley apparatus with the water extraction for 24 hours, and which is followed by 95% ethanol for 24 hours. And these rice extracts were preheated in the deionized water in the water bath 50 centigrade for 1 hour, 60 centigrade for 3 hours, and 70 centigrade for 5 hours, and 100 centigrade for half an hour. The whole process was done after the interval of 15 minutes. The 10 ml aliquot, which is obtained from the rice water extract, was then treated with a 2% H2SO4 and deionized water and then incubated at 100 centigrade. The extract was neutralized by using the calcium carbonate suspension. And how we determine the XOS, the clear liquid extract was filtered and placed in the ice bath. The autoclavable jar was placed in the autoclavable condition at 121 centigrade for 0.5 hours. The clear supernatant was collected, which is a rich source of oligosaccharide. and this extract was then inoculated into the overnight culture suspension of the lactic acid bacteria in the MRS medium and then incubated at 37 centigrade for 24 hours. And the growth of these culture with rice husk extract and without the rice husk extract was shown by and checked under the UV spectrometer after the absorbance values, and the absorbance values obtained after the after the 2 hours, 4 hours, 9 hours, 10 hours, you can change a potential increase in the absorbance of the culture 1 and culture 2, and the culture 2 contain the rhizus extract but the culture 1 does not contain the rhizus extract, and the highest peak value after the 18 hours were 0.806, which shows the highest absorbance values, which shows that there was a lot of growth after the 18 hours, and this is the growth curve which was obtained at the OD 600 nanometer. And how the rice husk was quantified. The rice husk was quantified, extract was quantified for using the rice extract was quantified using the HPLC, high profile liquid chromatography, and the chromatogram which was obtained after the chromatography contained that different have shown that different components in the rice extract, and these components were the xyloligosaccharides, which was the 68% in the rice husk extract obtained after this process, after my research, and the other second component which was arabinose, and the third component was sugar, was the sugar glucose, and the fourth component is xylanase. Xylanase was an enzyme which is used as a component to convert the other xylem resources like the uh, corn cob which is converted, which contain the xylem and which can be converted into the xylo-oligosaccharide, which is a prebiotic which I have obtained from the rice husk and can also be obtained from the corn cob by using the xylanases, which was a component of the my rice husk extract. So this rice husk extract is not only used as a prebiotic resource but also as a source of the xylanase enzyme, which can be further used to convert the corn cob and other biomasses as a renewable resource to convert and obtain different beneficial products. And these rice extracts also contain the phenolics which can be purified, and some unknown product which were 2.7% which can also be purified, and these were used in the juices as well as in different cards to increase their probiotic probiotic nourishment. And so this is how I have used the rice husk which were used to discarded in Pakistan, and now these were the renewable resources to obtain the prebiotic. And that's all about my research. Thank you for your attention.
Thank you, Amar Palvasha, for your informative talk. Now I would like to invite Sidra Bukhari from Qaid-e-Azam University, Islamabad, to present with title Role of Doxorubicin coupled nanoseria in modulating TRAIL and NF-kappa B markers in leukemic rat model. Sidra Bukhari.
Hello everyone, my name is Sidra Bukhari and I am a PhD research scholar in Department of Biochemistry, Qaid-e-Azam University, Islamabad. Our research interest includes the identification of different natural and synthetic compounds that shows anti-cancerous and therapeutic potential against leukemia. The title of my presentation is Role of Doxorubicin coupled nanoseria in modulating TRAIL and NF-kappa B pathway in leukemic rat model. Nanomedicines are developing as one of the new treatment options against multiple diseases. This study was designed to explore the anti-cancerous potential of nanoseria that are commonly known as cerium nanoparticles, and this combination we call We have coupled the nanoparticles that are serum oxide nanoparticles. We have coupled them with a chemotherapeutic drug, that is doxorubicin, and we call this combination as a nanomedicine. We have tested the efficacy of this drug against acute myeloid leukemia in Sprague-Dawley rats. Uh, Two signaling pathway that we studied in this study was trail and NF-kappa B pathway. Trail pathway is a apoptotic pathway that induces cell death in cell, while NF-kappa B pathway is a anti-apoptotic pathway that prefers cell survival. So this was the overall introduction of our study. Moving towards the methodology. First of all, we synthesized the cerium nanoxide nanoparticles. Then we coupled these cerium oxide nanoparticles with doxorubicin, that is our chemotherapeutic drug. And after that, we have done the characterization of these nanoparticles by different methods. Then we induces leukemia in Sprague-Dawley rats, and we divided these Sprague-Dawley rats into 5 different groups that are shown below. And these groups are normal, leukemic, nanoparticle, doxorubicin, and at the last, that is our nanomedicine nanoparticles that are loaded with doxorubicin. After the dissection of all these groups, we move toward the hematological studies. We have done hepatic and renal marker profiling. We studied the analysis of apoptosis through an annexin 5 assay. And for the gene expression studies, we extracted the mRNA from the tissue, then we synthesized cDNA against those mRNA, and then we had done real-time PCR. So these are some of the results. In the figure 1, panel 1, you can see that it shows the XRD analysis of our different groups. Then in the panel B, we can see that it is the UV absorption spectrum that shows the significant peak was obtained at 320 by our nanomedicine. In the figure in the panel C, you can see the results of FTIR of all the 3 therapies that we have done. And in the panel D, that is the emission spectrum of our therapies. In the panel E, that is shown here, and you can see that these are the SEM micrographs of our 3 different experimental groups. So this was the characterization of the nanomedicine that we have performed. In the figure 2, you can see that this is the DLS in which we measured the size distribution, average size, and zeta potential of our therapeutic drug. And after these characterization, we move toward the results that we obtained from the our rats groups. And in the figure C, you can see that this is the relative organ weights of liver, heart, and kidney, that you can see in panel A, B, C, and A, B, and C. In the panel A, this is the figure of a liver, and you can see that in nanomedicine that is shown at the end, the nanomedicine was the one, and in the heart, the nanomedicine was the therapy that reduces back the level of organ weight of liver and heart. So the nanomedicine in this case manages to lower the weight of liver and heart that was increased in the case of leukemia. This is the figure of blood parameters. You can see that in panel A, the results of WBC, lymphocyte, eosinophils, neutrophils, monocytes, and hemoglobin are shown. And these blood parameters were measured in every group in our 5 groups, 5 experimental groups. And you can see that the nanomedicine and these 3 therapies manages to revert the condition back to the normal. Like in the case of WBC, the level of WBC in leukemia was increased, while the nanomedicines and nanoparticle doxorubicin and our nanomedicines manages to revert back the condition of WBCs back to the normal. So by calculating these overall blood parameters, we came to the conclusion that the nanomedicine was the drug that significantly reverted back the condition back to the normal. In the figure 5, these are the hepatic and renal biomarker results, and we can see that the nanomedicine against reverted back the condition of rats back to the normal in ALP, AST, ALT, and also the level of creatinine. So in case of leukemia, the level of ALP was reduced while the nanomedicine reverted back to the normal. In same is the case with the AST and ALT, the level was increased in case of leukemia, and our nanomedicine significantly reduces the level of AST and ALT in different rat groups. And the level of creatinine, it was also increased, but the nanomedicine and the nanoparticles both manages to reduce the creatinine level in rat groups. So in figure C, you can see the percentage of apoptosis that we measured through an XN5 assay. And you can see that in the normal group, the apoptotic cells, or the percentage of apoptotic cells was 4%, while in leukemia it was 77%. And this apoptosis was increased in case of our nano group, that is nanoparticle, sodium oxide nanoparticle, and doxorubicin. So this nanomedicine increases the percentage of apoptosis in AML cells. Moving toward the gene expression studies of our 2 signaling pathway, TRAIL and NF-kappa B pathway. This is in the figure 7. You can see the results of gene expressions of TRAIL pathway. This is the DR5, which is the marker of trail pathway, and a CFLIP, that is the inhibitor of trail pathway. We can see that the level of DR5 is significantly reduced in leukemia, but our nanomedicine completely enhances the expression of DR5. Same is the case with the CFLIP, that is the inhibitor of trail pathway. The inhibitor of trail pathway in the case of leukemia was highly increased, while our nano drug manages to decrease the expression of CFLIP, that is the inhibitor of trail. And the caspase 8, that is really important for the induction of apoptosis. We can see that only our nanomedicine, that is the nanoparticles coupled with doxorubicin, manages to increase the expression of caspase 8. So all of this study shows that the nanoparticles not only induce apoptosis, but it triggers trail pathway for the induction of apoptosis by increasing the expression of DR5 and decreasing the expression of CFLIP. Same is the case with the NF-kappa B pathway in the figure 8. You can see that we have used 2 markers, REL-A and REL-B, for the study of NF-kappa B pathway. And for both the markers were increased in case of leukemia. When this marker was increased in case of leukemia, that prefers the cell survival, but the nanomedicine manages to revert back the condition or down regulates the expression of REL-A and REL-B, which shows that the our nanomedicine inhibits the NF-kappa B pathway by down regulating the REL-A and REL-B pathway. So at the end, our results shows that the sodium oxide nanoparticle may enhances the apoptotic potential of doxorubicin both at cellular and genetic level. And we have also studied that it also inhibits the pro-survival NF-kappa B pathway markers as nanomedicine, and it also rendered itself as a potential therapeutic agent against acute myeloid leukemia. So this was our all study against cerium nano-oxide nanoparticles for the to prove that they can be a good therapeutic marker for the study of leukemia, specifically acute myeloid leukemia. Thank you so much for your attention.
It was an amazing talk by Sidra Bukhari. Now, our next talk is by Dr. Azhar Rafiq from Government College University, Faisalabad, with title Efficacy of Anthelmintic Properties of Leaf Extracts of Moringa oleifera and Azadirachta indica against nematodes of Ovis orientalis orientalis. Dr. Azhar Rafiq.
Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Azhar Rafiq, Assistant Professor, Department of Zoology, Government College University, Faisalabad. The topic of my presentation is efficacy of anthelmintic properties of leaf extracts of Moringa oleifera and Aza directa indica against nematodes of Ovis orientalis. First of all, the introduction. Helminths are the parasitic worms that feed on a living host to gain nourishment and safety while inflicting terrible nutrient absorption, weakness, and disorder within the host. Sheep gastrointestinal parasites are of public health significance and they are incriminated in zoonotic transmission to human. Mufflon sheep is an animal species that is widely hunted and produced in Central Asia. Mufflon sheep are very sensitive to many parasitic diseases, especially fasciolosis, nematodiosis, etc. These diseases cause health problems and animals die prematurely and cause economic losses to breeders. Chemotherapy is a chief remedy modality used for helminths infection in animals. However, because of increasing improvement of anthelmintic resistance, phytotherapeutic tablets are secure, non-poisonous, biodegradable. The consequences recommend that the phytochemical drugs for using diverse illness. Moringa leaves and stems contain amounts of bioactive compounds. All parts of the neem plant, including the leaves, bark, fruits, seed, oil, and sap, have been shown to have medicinal properties and contain over 10 different active components with azadirachtin as the most potent and widely studied. Then the materials and methods. And the preparation of, first of all, the preparation of plant extract, collection of leaves of Moringa oleifera and Azadirachta indica, then dried at room temperature, ground into fine powder and extracted with aqueous methanol and ethanol solvents. Egg hatch assay, aqueous methanol and ethanol extracts of both plant Moringa oleifera and Azadirachta indica were used as the test treatment. Fecal samples of mufflon sheep were collected at the wildlife park at Faisalabad, Pakistan from the ground after defecation. Approximately 9 to 110 eggs of nematodes in 0.5 ml water placed in each test tube. Serial concentration of each plant extract in total volume of 0.5 ml in distilled water was added to make concentration of 50 to 1.5 milligram per ml together with water containing the eggs. The test tubes were then covered and kept in incubator at 27 degrees Celsius for 48 hours. The experiment was conducted in duplicates for each concentration and replicated 3 times. Hatched larvae, dead or alive, and unhatched eggs were then observed under dissecting microscope at 40 multiple magnification. Then the larval development test. The experiment was conducted in plastic cup of 20 ml according to the technique described by Costa et al. An aliquot of 1 ml containing 100 to 121st stage larvae of nematodes were mixed with 5 grams of feces from a sheep free of gastrointestinal nematodes and 6 serial concentration of the plant extracts with water containing larvae and volume of egg free of feces. All samples were incubated for 7 to 14 days at room temperature. The experiment was conducted in duplicates for each concentration and replicated 3 times. Then 1 drop of Ligol's iodine solution was added and all L3 stage larvae were counted under dissecting microscope at 40 multiple magnification. For positive control, ivermectin 1% was used while negative control, untreated eggs and water were used for both tests. Then the results, this table shows the egg inhibition, egg hatching inhibition percentage and LC50 of 2 plant extract compared to negative and positive control. And the all plants showed egg hatching inhibition. By increasing concentration of extract, the egg hatching inhibition also increased. The maximum egg hatching inhibition was 88% in ethanol solvent of Moringa oleifera, while the maximum concentration of egg hatching inhibition was 94% in methanol solvent of the Azadirachta indica. These are the values of the LC50 of Moringa oleifera in aqueous methanol and ethanol solutions. And these values were the Azadirachta indica in aqueous methanol and ethanol solvents. This table shows the development of the larval inhibition percentage and LC50 of 2 plants extract. compared to negative and positive control. Both plant extracts showed larval development inhibition. The maximum inhibition was observed in methanol as a solvent of aza directa indica. These values were LC50 of Moringa orifera in aqueous methanol and ethanol solution, and these values of LC50 of aza directa indica in aqueous methanol and ethanol extract. It was observed that methanol extracts of aza directa indica is the most effective against the nematodes. Then the conclusion from the present in vitro study, it is concluded that ethanolic extract of Moringa orifera and ethanolic extract of aza directa indica exhibited significant enzymatic activity against nematodes of sheep and have potential for use as an alternative enzymatic agent for the control of gastrointestinal ailments of wild sheep and other ruminants. The two plants in our study were effective in the treatment of gastrointestinal nematodes of wild sheep. However, it would be necessary to conduct in vivo parasitological studies to consider the metabolism of extract in digestive tract of ruminants in the captivity. Thank you for your attention.
Thank you, Dr. Azrafiq, for your nice talk. Coming towards our last talk by Dr. Saima Sharif from Lahore College for Women University, Lahore, with research topic Allergens and Food Allergens Sensitization Patterns in Young Adults. Please, Dr. Saima Sharif.
Bismillahirrahmanirrahim. I'm very thankful to the third organizers of Third International Conference on Applied Zoology that they have given me the opportunity to present my research work. I'm Dr. Saima Sharif from Department of Zoology, Lahore College for Women University. The topic of my presentation is Aeroallergens, Food Allergen Sensitization Patterns in Young Adults. Allergen sensitization is a strong risk factor for the development of respiratory diseases, and it's the allergen sensitization occurs when suspectable individuals are exposed to allergens, as you can see in this diagram. Once they are exposed, then definitely that affects the lungs and the respiratory tract. So aeroallergens are airborne particles that induce allergic reactions in sensitized subjects and cause respiratory allergy. Examples are the dust mites, cockroaches, pests, molds, and outdoors like pollens or molds. Food allergens are proteins or glycoproteins that produce malfunctioning of the immune system in response to the dietary antigens. Examples, milk and dairy products, eggs, peanuts, nuts, and gluten-containing cereals. There are very simple tests that was performed to detect the aero allergy or the food allergy. They're the two simple tests performed for this purpose. The skin prick test, during which a small amount of substance was inserted by a prick near the wrist, and if a reddish elevated bump with red ring appears around it, it will indicate the sensitization to the allergy. And second is the immunoglobulin test, that is the determination of immunoglobulin E by ELISA in the blood samples. For this small study, we have taken 500 subjects of various age groups from the different laboratories of Lahore. Then the blood was collected and subjected to the ELISA for the determination of the immunoglobulin E. The data was analyzed to check the frequency and percentages of the allergen sensitized subjects. Definitely, we analyzed the frequency of the substances, either they are having the aeroallergy or not. So the total population was divided into the 5 groups on the basis of age. That was from year less than 1 year, from 1 to 5 years, 6 to 10 years, 11 to 15 years, and above 16 years of age. The mean study population have the mean age of 34.76 years, and the mean immunoglobulin that was being determined in this age group in our whole population was 2,595.15 international units per ml. And it was observed that most of our studied group were having were not having normally required values of immunoglobulin and hence are susceptible to aeroallergy and the food allergy. In this tables, we have mentioned the reference ranges of immunoglobulins that from 0 to 1 year. The reference range is 30 micro international units, but we determined that in our collected samples, the immunoglobulin levels was 189, which were much higher than the recommended reference range. Similarly, in the age group 1 to 5 years, that was 434, and it was above the reference range. So if we find out the frequencies, that how many number of subjects are having higher immunoglobulin levels, we find out that in the age group 0 to 1 years, 75% individuals are allergen sensitized. In group 1 to 5 years, 72% are allergen sensitized. In 6 to 9 years, 87.5% are allergen sensitized, and in the group 10 to 15 years, 57.14 subjects are allergen sensitized. And in the age group that was considered as adult, that was above than 16 years of age, 67.28 number of subjects were. We have calculated the percentage of that how many subjects are being allergen sensitized. So this is an alarming condition that in adults, that was 67.28% that are being affected, and even in the children we observed that 87.5% that was a high risk that the subjects or the children are being allergy sensitive, or they are being having the allergic responses. And if we further categorize the subjects which are above than 16 years of age, we observed that the subjects of age 16 to 30 years, there are 130 subjects which are being affected. So that indicates that allergic responses was very much present or prevalent in the young population. So why this food allergy or air allergy is increasing in adults? It was maybe one of the main reason is that Lahore is a very populated city, and it has pollution risks also, and also the increased exposure to poor quality of air that may causes immune reactions in that. And if immune system cannot outgo allergies, then the sensitized individual carry these allergies throughout the life, and such adults are the major part of the population having food and air allergies. Moreover, adults of active lifestyle group can't avoid such foods which causes allergic responses, like most of the fast foods which are present and which becomes the habits of our daily use because of their busy schedules, traveling and other meetings. So it was concluded that there was very limited studies in Pakistan that bring the consistent issue on frontline, which would be addressed permanently at the public health level, as 72% children and 67.28% adults were allergy sensitized. There is need to improve the indoor and outdoor air quality, which is playing a major role in exacerbating the allergic diseases upon exposure. So this our brief study recommends detailed research on the general population to analyze the allergy patterns, complications, and real trends related to allergy. Continuous and excessive exposure to aeroallergens that trigger the immune response should be minimized, and dietary allergic products and food intolerance should be avoided. Minimize your exposure to pets and smoking. A healthy lifestyle and clean hygiene are highly recommended to boost the immunity. Thank you so much.
Thank you, Dr. Saima Sharif, for your informative talk. So our fourth session of day one has been completed. I'm thankful to all the viewers, researchers, and faculty members who were engaged throughout the session. Our next session will start at 5 p.m. Sabaryaz will be the moderator of our fifth session. The session will comprises the talks from the fields of ecology, biodiversity, fisheries, wildlife, and nutrition. Stay connected, stay tuned. Thank you.